most of the talks in population biology, but I hope there is some overlap that we can discuss. Both meetings being recorded. And happy Earth Day. It's really cool to be giving a talk about my research, which I hope has some potential to help contribute to a better Earth in the future on fantastic Earth Day. Although I will admit, as beautiful as it is outside, my Canadian heritage means I am worried about melting. It's really, really warm for me. So I want to talk not about my dinosaur research, but actually about my mammal research today. And I want to put forward an idea about how we might be able to integrate some paleontological data with modern data to better understand the rules that govern evolution in these animals on long time scales, and hopefully use that kind of as a long-term impetus to try to figure out better ways to conserve the biodiversity that's around us. So those will be the kinds of themes that run through my work, and where I'm really interested to get some discussion going about how I can possibly do that, because coming from a paleontological background, of course, I work with data in different ways than all of you do, and so I think the cross-pollination could be really fruitful. So I want to talk today about mammal life history and the evolution of life history. So to get on the same page, I want to talk first about what life history actually is. There we go. The life history of an animal is the pattern and pace at which it progresses through the different phases of its life, from birth or hatching through childhood to its reproductive adult form. And it's long been known that life history helps to govern the ways that organisms interact with their ecosystems, and it's implicated as a major factor in extinction risk. And so life history has become this central topic in conservation biology. And when we look at that diversity of tetrapods around us today, we see a myriad of different life history styles. I'm interested in how these different life histories evolved in these animals, and what impact life history has then had on the subsequent evolution of these lineages through geologic time. These are central questions for understanding the trajectory of life on Earth and the resiliency of species to extinction, and understanding and answering them requires investigating the life histories of extinct organisms. But how do we study life history in the fossil record when all that we're left with, if we're really lucky, are little bits of bone and teeth like this? This is where we turn to an approach called paleohistology. This is the study of fossilized tissues, and this has become a main aspect of my research on both dinosaurs and mammals. To reveal these tissues, we make a thin section of a fossil to look at it under the microscope and shine light through to reveal the tissues. We start by cutting it very carefully with a specialized slow spinning saw. And then we grind and polish it, either by hand or using abrasive wheels that allow us to precisely control the thickness of the slide. And this is a grueling process, as you might be able to tell. And ultimately, we end up with a slide that is thin enough to allow light to shine through it. And this reveals the microstructure of the fossil. And this microstructure can provide a wealth of data on life history. Numerous studies in this field now show that the original cellular textures of the skeleton are preserved through the fossilization process, and that these microstructures reflect the rate at which the skeleton was deposited when it was being formed. Tissues laid down more quickly tend to be more disorganized, like this woven bone on the left, whereas slower deposition results in more organization, like the tissues on the right. And so this can tell us, in general, the rate at which the skeleton grew, kind of on a gradient between fast and slow. But on top of this, as bones and teeth develop, they're affected by cycles in metabolism, from daily oscillations in the circadian rhythm to yearly photoperiod sensitive hormonally mediated cycles, basically summer-winter cycles in our lives. So on the left here are some daily cycles of growth in the enamel of a mammal, whereas on the right are some annual cycles of growth in the pubis of an oviraptor or dinosaur, one of those toothless dinosaurs I worked on for my PhD. And so these cyclical growth marks give us a clock to understand the way that growth rates changed over an organism's lifetime, and to potentially plot out some information about their biology over the course of that chronology. So in addition to anatomy, phylogeny, and macroevolution, my research uses these kinds of paleohistological techniques to better understand life history and growth in fossil vertebrates. I'm particularly interested in understanding how growth patterns have changed over time, as well as what effect they've had on the evolution of these different lineages. And one of the best ways to understand how life history affects evolution is to look at when things go wrong, and especially to look at things like extinction events, which reset ecosystems and allow us to see what evolution does afterwards. And so I've been looking to extinction events in the past to understand how life history affects survival through extinctions and radiations afterwards. My current research is centered around the end Cretaceous mass extinction, which is dramatized here. I imagine some of you will probably be familiar with this extinction, but as a recap, a bolide or meteorite impacted Earth, collapsed ecosystems, and resulted in the extinction of the non-avian dinosaurs. The birds survived around us today, including the peregrine falcons, which are up on the water tower by EPS. That's always cool to see. It might also be familiar that afterwards, in the aftermath of this impact, mammals rose from the ashes, as we like to say, to take over the niches that were once occupied by the dinosaurs. And they established the first mammal-dominated ecosystems that are the precursors of what we have around us today. And this is the first time in their history that mammals got bigger than the size of a badger. And they did it in just a few 100,000 years. And it wasn't just any mammals that got big. It was the Eutherian mammals, our ancestors of the placentals, that got big in particular. And this is interesting because these mammals were not the dominant group in the Cretaceous. They were kind of stragglers, and there were other mammal groups that were more common. And this paradox has long plagued paleontologists. Why did the Eutherians take over, and what enabled them to get big on these really fast timescales? I've been interested in seeing whether life history, particularly reproduction, played a role in this transition. And there's been this loosely held idea, if we look at a phylogeny of mammals around us today, the Eutherians took over because they have distinctive life history strategies, specifically their maternal investment strategies, how the mothers take care of their young. And there's this idea that placentals were exceptional, because if we look at modern groups, only the placentals have precocial babies. They have the capacity through a more tightly integrating placenta to produce precocial babies through longer gestation periods, whereas marsupials and monotremes either lay eggs in the case of monotremes, or they give birth to tiny little babies that live in a pouch for much of their development. And so there's this idea that that trait of placentals is what made them exceptional and allowed them to radiate after the dinosaurs went extinct, that in some way they were superior to other sorts of lineages. So it's important to see if we can determine the life histories of the organisms that lived just after the extinction to see if this trait actually evolved in the Eutherians at this time, or if this was something that evolved later on, and to test if it could have actually played a role in this diversification. And one of the best places to study this is the traditional territory of the Ute, Pueblo, and Navajo and Jicarilla Apache peoples in the San Juan Basin of northwest New Mexico, where I gather we have some people in the room from. Maybe not right here, but New Mexico itself. So rocks in this area extend through the Cretaceous and Paleocene era, and they preserve a rich assemblage of fossil mammals, specifically the teeth. In 2022, my co-authors and I had a pretty big paper come out that used some of these teeth to get a new window into the life history of these extinct animals. And that's what I'd like to run you through today, and then get into a little bit of where my future research is going to go. The approach that we used relies on creating thin sections of these fossil teeth to reveal those daily growth lines that are preserved in the enamel. And so I've highlighted, this is the tooth before we thin sectioned it. This is it afterwards using a type of light called cross-polarized light that's common in mineral petrography. It basically shows you the refraction of light through the tissues. And I've highlighted here the enamel, this kind of bright tissue on the outside of the tooth. And mammal enamel provides a wealth of life history data because of those key structures that I talked about. Mammal enamel is made up of enamel prisms, and these are basically tracts of enamel that are deposited by a single ameloblast cell as it travels from the dentino-enamel junction outwards towards the outside of the tooth. So basically, each of these vertical lines is produced by a single cell that leaves behind enamel through its growth. And I like to think of it kind of like a jellyfish. It kind of pulses along each day, and as it does, it leaves behind a tiny little bleb of enamel separated by a rest line. And that rest line is called a cross-striation. It essentially marks the circadian rhythm when that cell is producing enamel versus when it's producing less. And these growth signals are particularly useful in mammals because the prisms are well developed so we can see them quite clearly. But we also have multiple ameloblasts producing enamel at any one time. And so you have a growth front that manifests as these laminations. So they basically represent the shape of the tooth on a particular day. And then you have all of the ameloblasts pulse up and they advance the tooth. And so you can see these growth fronts of the tooth and reconstruct them in these fossil organisms. But it's not just this daily chronometer that's interesting and important for us. I guess I should mention here, the reason these arrows are here is that you have growth in two directions. So you have growth of the ameloblast through its lifetime up that way, and you also, because of the angle of the tooth and the way that teeth develop, you have advancing growth fronts that extend the growth in this direction. So there's two kind of directions of growth going on in this tooth. But in addition to these daily signals, teeth can also preserve really interesting growth marks. This is an example of the birth line. So because our teeth start to form, even in utero, even our adult teeth, they can record this birth transition. If you've ever seen an X-ray of a toddler's skull, all of their teeth are in there, it looks kind of gross. This is the benefit of that kind of thing. Our teeth start to form in utero, and so we start to deposit enamel while we're in the womb, and then there's a transition when we're born. So this birth line is represented by a seam in the enamel prisms that reflects the stress of birth. Most parents in the room will know that when you give birth to a child, that child tends to lose weight in the first few days and then start to recover and grow afterwards. And this is basically the reflection of that in the skeletal tissues. Now, birth lines have actually long been known in living animals, but they haven't really been recognized as much in fossil organisms, although they are relatively easy to identify. Now, even though they're identifiable histologically, we do want a little bit more evidence of what this transition actually is. Could this just be some other sort of stress in this animal's lifetime? And what's really exciting about our study is that we actually have corroborating evidence from an independent line of evidence from geochemistry. And this is because of a unique feature of mammals. All mammals are united by the production of milk, but they rely on it to different degrees. This is that maternal investment dichotomy that I talked about. For monotremes, like the platypus and marsupials, which is that guy, not that one, it's a crucial part of their development because they give birth to these tiny little jelly bean-sized babies that crawl up to the pouch and latch onto a teat semi-permanently, and then they undergo most of their development early on in the pouch outside of the mother's body. And so they rely on that milk coming from the teat during that interval. In contrast, placentals like us, we actually go through more development in the womb, and so we tend to rely on milk for a much shorter period of our development after we're born. Now, milk leaves actually two unique biochemical signatures in the teeth, and that's because there are actually two types of milk. The first is colostrum, and if there are any mothers in the room, you may know about colostrum. It's produced only for the first few days after birth, and I like to think of it like rocket fuel. It's basically supercharged with nutrients and antibodies, and it's critical in developing the young's immune system, giving it a head start in life. After this, the mother will produce a more dilute breast milk, which is more like a balanced meal, and it provides most nutrition for the young early on in their life. And these two types of milk correspond to different trace element signatures. So colostrum is super enriched in zinc. Zinc is this kind of do-all nutrient that's really important in a lot of different body processes, and because this colostrum is enriched in zinc, our bodies become enriched in zinc when we're drinking colostrum early on in our lifetimes. The other element is barium, and barium's uptake is a little bit more complex. Digestion of casein proteins in milk produce phosphopeptides that enrich the absorption capacity of calcium. And calcium and barium have a similar valence, and so barium kind of acts as a hitchhiker in this process, and it gets incorporated into the hydroxyapatite structure. And what's really interesting is that because of the way this process works, even if you're not ingesting more barium, because it becomes more bioavailable in the presence of milk, you absorb more barium into the teeth during this period of breastfeeding. So when you're drinking milk, you'll absorb more barium into your skeleton, as well as calcium. Now in humans, of course, this doesn't work because I had a latte this morning, but most animals, as far as I can tell, don't do that. There are some organisms that have periodic suckling and weaning, and we can actually see that signal in the teeth. We can see those annual or semi-annual bands in things like orangutans and possibly Neanderthals as well. Now, we should be able to use these two signals of different phases of milk ingestion as signals of critical periods in life. So zinc should be enriched just after birth when the young is drinking colostrum and in the skeletal tissues that are formed right after birth, whereas barium should be enriched throughout the suckling period when it's increased in that bioavailability through the consumption of milk. And we can reveal these patterns in our fossils by zapping them with a very fancy laser. We used a technique called laser ablation inductively coupled plasma mass spectroscopy. or LAICPMS for short, which isn't really that much shorter. And we used a fantastic rig up at the University of St. Andrews. And this is my colleague Jacob, who's now actually at the University of Washington, loading our specimens here, one of those little thin sections, into this chamber for vaporizing. That vapor gets sent to a mass spectrometer, and this measures the concentration of the trace elements. And this technique of using LAICPMS for geochemical analysis of fossils is actually becoming pretty common. But we used an unconventional technique where instead of doing a spot sample, we sequentially rasterized the entire sample. And we used a time-resolved analysis to match up the peaks and troughs of our geochemical signal with the position of the laser at the time when we were scanning it. Now, this allowed us to look at these biogenic trace elements, things like zinc and barium, but it also allowed us to look at the byproducts of fossilization, things that we would call diagenetic minerals or diagenetic indicators. These are introduced into the skeleton when that fossil is stabilized during the fossilization process. And this is what that looks like in action. This is kind of like a really crappy record player where you're essentially dragging this laser as though it's the needle of the vinyl across the specimen, and you get these peaks where you can see what's going on in the specimen. Now, in our case, we were able to do this at really high resolutions. So the resolution of the laser spot and the way we're dragging it across the sample meant that we got resolution of about 60 microns squared. To put this into context, this is about 3 pixels per day of growth in this animal in the short direction. So it really gives us high resolution to see what's going on at an unprecedented level. So here's an example of what this looks like. We're going to look at a paraconid of a second lower molar of an animal called Pantalambda, which we'll become more familiar to in a second. And this has a great growth record because in this tooth, which I'm showing here, we've got the cusp of the tooth, which is the place where the tooth formation initiates. It grows down in this direction, as well as slightly out in this direction. But this tooth has a birth line preserved here, which we identified histologically. And it then has this long sequence of enamel developing afterwards. So it has the potential to capture this entire window of this animal's early life. And when we look at the zinc heat map, this is what that looks like. And for me, this was really a kind of eureka moment where we suddenly realized that this has actually worked. So what we see here is we can see some signals that we were expecting in these teeth. So for example, zinc is enriched at the surface of the enamel, and that actually helps to protect the tooth in the oral environment, to protect it from dissolution. We also see this little line just here that matches up exactly with the histological birth line. And this, we think, reflects the ingestion of colostrum early on in this animal's life just after it's born. And this kind of signal is exactly what we see in modern teeth when we do the same thing. It's not just in this one cusp of the tooth. We looked at the other cusps of the teeth that have birth lines as well, and they show the exact same signal. So the birth line does appear to be enriched in zinc in our fossil animals. When we look at barium, we do see that same expected pattern of high barium early on in life, followed by a period of low barium when we suspect that the animal has weaned and it's not drinking that milk anymore. Now, it's important to reiterate that these are maps of a fossil that is 63 million years old, but we're getting the same kind of precision and results that we would get in a modern organism when we do these same sorts of approaches. And to me, this was really fantastic. This gave the idea that not only are the trace elements within the enamel preserved with high fidelity through time, but their spatial arrangements within the tooth are as well. And that's really exciting to see. So this could become a really powerful approach for understanding reproduction in these extinct animals. So we can identify birth and the suckling period to the day because we have those daily growth marks through our tooth. And we can correspond those two, we can correlate those two signals, and we can register the trace element map, and we can actually count out how many days this animal was suckling for and how many days before it was born the tooth was developing for. So this allows us to estimate gestation and suckling periods to the day in these fossil animals. And this is important, especially because remember that a lot of our modern mammal groups rely on milk to different degrees and versus maternal investment. So this is a major signal of that dichotomy in maternal investment that we have today. So to put this in a different sort of visual way for you and to summarize it, we can use the daily increments in the tooth to produce a daily timeline of this animal's life. And then we can use both histological and trace element signals to plot the positions of particular life history events onto this daily chronology, sorry, that's the word, this daily timeline. And this allows us to estimate the length of the gestation period and the length of the suckling period to the day, as well as some other features later on in life. I won't have a chance to talk about those today, but we can get a lot of information out of teeth. Now, when we applied this to Pantolambda, and all of the data that I showed you comes from Pantolambda itself, when we align the trace elements to that chronology, we get a very distinctive Eutherian-looking signal. So we have a long gestation period of 207 days, just under 7 months, and a suckling period, depending on the individual, of between 1 month and 2 months. And the precision of these measurements on the order of days allows us to directly compare this fossil animal 63 million years ago to the organisms that are living around us today, which we can observe these patterns in. The gestation period of Pantolambda is exactly in the zone we would expect for a living placental mammal of its body size. So right up in this zone here, nearby some other potentially familiar organisms there. And this is an order of magnitude longer than what we see in marsupials. And so it suggests that Pantolambda, even in this early phase of Eutherian evolution, had the right sort of internal machinery to gestate the young for a long period. Although we can't necessarily say that it had a particular style of placenta. This analysis doesn't reveal those soft tissues. When we look at its breastfeeding period, Pantolambda seemed to have weaned fairly early, but this makes a lot of sense in light of a longer gestation period. And this leads to a well-developed baby with less reliance on milk after it's born. And when we incorporate these reproductive characteristics alongside some other life history signals that we evaluated histologically, but which I don't have time to talk about today, unfortunately, we find that Pantolambda is solidly within the life history realm of placentals. Specifically, it plots close to many highly precocial ungulates, things like the antelope and the dik-dik. And these give birth to relatively well-developed babies, a single one usually, that is mobile within hours after birth. They're up and running around. The difference is that Pantolambda is much larger in body size than these nearby species. And this suggests it has a relatively fast life history or pace of life for its body size. But again, importantly, it's quite far away from our non-placental mammals, like marsupials and monotremes. And this suggests that even during this early phase of their evolution, Eutherian reproductive physiology was distinctive. It was quite different from what marsupials and monotremes have today. But of course, this is just one species. Pantolambda is one member of a lineage which has no living descendants right now. Not right now, it just has no living descendants. Until we figure out where it goes in the tree, I guess. Maybe it does. But it might not actually be representative of all placentals in its ecosystem. And so the natural next step is to look at the other contemporary mammals that lived around Pantolambda and see if we can get the same kinds of signals from them. So I've got a sample now that has 16 different taxa, lots of teeth, lots of bones, try to get these life history signals spanning basically the earliest part of the Paleocene, from about 66 million years to about 62 million years-ish, so that first 4 million years. And I've also just received a whole bunch of new geochemical maps from some key taxa within these. And what's really exciting is that, first off, what we find is that the signals we were finding in Pantolambda are not just one-offs. They didn't rely on any sort of spectacular preservation. Not that the Pantolambda teeth were spectacularly preserved. They're the kind of teeth that I was allowed by a collections manager to cut up in an experimental fashion. So these were not necessarily fantastically preserved teeth. But what we find is that in every single specimen, patterns in our diagenetic indicators are different from those that we see in our biogenic indicator minerals, our trace element signals. And so this supports the idea that enamel really is a vault that preserves these signals over long time periods, and that we might be able to generalize this method to look at broader patterns, not just in these Paleocene things, but even further back into the past. And for me specifically, it's really promising because it gives me hope that my entire research program might actually work. These analyses also reveal some weird things going on that we weren't quite expecting. So here's an example of one of the quite interesting signals that we have. If any of you have looked at lithium in your living organisms and have any sense of how it's incorporated into the skeleton, please let me know, because there's no literature on this. Outside of looking at antidepressants and the placental boundary, lithium and its incorporation is really not well known in a lot of organisms. It's not usually naturally uptaken. But what we see here is that lithium is definitely different during different phases of this organism's life. We're not really sure if it corresponds to a birth transition or a suckling transition, although my guess would be based on the barium, it corresponds to enrichment during suckling. But of course, showing that would require a modern sample where we can better understand and control these ideas. Here's another example where we see some other things going on in lithium, but in this case, it might not necessarily be related only to suckling, because we have a birth line right here. And what we find is that lithium is enriched on both sides of that, but it's not enriched throughout the entire lifetime of this tooth. So there's something going on here with lithium that we need to figure out. But in any case, I basically preliminarily crunched these data from these new specimens that I've got, and I've incorporated them into that same analysis with Paleolambda. And what we find is that placentals really did run the gamut of life history styles early on in the Paleocene. They did seem to have basically a representation of the diversity they have today in terms of how they're investing in their young. Even some that seem to be kind of verging on the overlap between marsupials and placentals. So maybe some interesting things to go on there in terms of extinction dynamics and recovery dynamics. So in any case, this combined paleohistological and geochemical approach has given us a new window into the life histories of these early mammals and the early parts of their lives with precision that is similar to living mammals. And that, I think, is really exciting. And perhaps most importantly, the thing that I'm most excited about is that it's viable even in these 62-million-year-old fossils. And to me, this opens the door for similar investigations further back in time into mammals on the other side of the KPG boundary. And that is exactly what I'm currently working on, building from my Banting Fellowship at the Royal Ontario Museum, where I met Luke Muller, who suggested I do this talk. Luke is a former CPB postdoc, spoke very well of UC Davis. And I've been sampling specimens from a fossil formation called the Bug Creek, part of the Hell Creek Formation. I'm aiming to answer 3 different questions. First, I'm testing the assumption that Mesozoic precursors actually had the same life histories as their living counterparts do, to evaluate if it's even actually feasible that it might have been involved in their survivorship and growth after the extinction event. And ultimately, this will let us test if placentals actually took over because of the extended gestation periods we see in things like Paleolambda and its contemporaries. So I've been studying the dental histology of the 3 major groups of mammals that existed at the end of the Cretaceous. We have the Eutherians, which I've introduced you to. We have the Metatherians, which are ancestors of marsupials. And then we have this other group called the multituberculates. Now, how many of you have actually heard of a multituberculate? We got 1, 2. Okay, that's great. Multituberculates got a pretty raw deal because they have the distinction of being the longest lived group of mammals. They have a temporal range of more than 130 million years, but they went extinct in the Eocene. And so we don't really have any representatives today. They were possibly outcompeted by rodents, although that's not fully refined yet. But they are a really important group because they also survived the Cretaceous extinction, and actually in the aftermath, they also got quite big, up to kind of the size of a beaver or something like that. Now, we've generally assumed, or at least paleontologists have generally assumed, that Eutherians and Metatherians in the Cretaceous lived like their modern counterparts do. That is, giving birth to well-developed young or giving birth to pouch young. But we don't really know too much about the multituberculates yet. They're key for kind of figuring out this story. But there is a fantastic paper by my collaborator looking at the growth of bones and using those dynamics to predict that they actually had more placental-like life histories. So basically what I've been able to analyze representatives of each of these different lineages, and I'm just going to run you through some of the preliminary results that we have on this to kind of give you an idea of what the story is doing at this point in time. So I'll start with the Eutherians, and essentially what I found is basically they're the exact same as what we find today or back in the Paleocene. Most of the teeth show beautiful enamel laminations, which you can see here. This is a tooth of a tiny little thing called Scalardius. Its skull would be about centimeter or 2 long. This is a really small tooth. Ask me about that over a beer if you're interested in how much of a struggle that is to dissection one of those. But what's really exciting is that these Eutherians also have birth lines. We can detect these histological signals. And this one is really exciting because it actually shows what we call the classic staircase morphology that we see in birth lines in the modern. So it's kind of got this You can see basically the tips of the prisms. Now, of course, we would like to do some geochemical work on this. These results are still preliminary. We haven't had a chance to put these under the laser yet. Lasers have a tendency to go awry and then stop working. But it's exciting that we already have candidates popping up in some of these Mesozoic fossils, that there's no noticeable appreciation of a decrease in preservation quality across this boundary. These things really do look like what you would see in a modern fossil or modern organism. So if I turn now to the metatherians, these are a really interesting part of the sample because except for some of Thoms's work early on in the 1800s, there have never been analyses of the teeth of these things histologically, as far as I can tell. If you have other references, I'd be really appreciative to hear them. Now, we also have quite a size range of our metatherians, which I've shown up in the top right there. So this jaw of Paleomus is tiny little mouse-sized. Didelphodon was about the biggest Cretaceous mammal that you can get. It was about the size of an otter, and it had these kind of domed, crushing teeth that we think it used in part of a Durophagous diet. Now, what's really exciting is that these seem to show the exact same sorts of incremental structures that we find in the eutherians. For example, with Paleomus, we've got a deciduous lower third premolar here, which is one of the early generations of teeth. And in our eutherians, our deciduous molars almost always have a birth line in them because they always develop or start developing in utero, unless you're a mouse, in which case they develop and then are lost before you're actually born. And we can see really nice enamel laminations here in this deciduous premolar. And when you count these up and you calculate how much of the tooth is missing and you calculate the time it took to form the tooth, it looks like it would have taken just over 2 weeks to form this tiny little baby tooth in this marsupial, which is kind of interesting to put it on that sort of perspective. Now, noticeably absent from our deciduous premolar is evidence of a birth line. And this suggests to us that these marsupials, at least tentatively, were probably not developing their teeth early on before they're born. And this might be a sign that these are living a more marsupial style life with more reliance on a pouch and a longer period of relying on milk. The multituberculates were really exciting because, again, nobody had really sampled these and they pose a major question. So here's an example of some of the size range that we've got. Again, quite a large size range. Meniscoesis is not quite as big as Didelphodon, but it is kind of opossum sized, so a fairly large multituberculate. So when we look at these teeth, what's really exciting is that we seem to get some potential evidence of a placental-like life history in them, which would fall kind of in accordance with what we've seen based on that long bone, the skeletal growth hypothesis. And so here's an example of the molar of meniscoesis, and we have these accentuated lines. This is in the dentine, not in the enamel, and this implies that this tooth was already in the process of erupting when these birth lines were formed. And we think maybe this is the birth line and this could be a weaning line. But again, it'll rely on geochemistry to kind of tease apart these signals and verify this, because we are looking pretty far outside of our extant bracket here in terms of what we can reliably infer. But it is exciting that in any case we have some candidates, and they seem to support previous ideas that these animals were a little bit more placental-like. And so this throws a bit of a wrench in things, because if we're looking now back at our tree of mammals, what we find is that yes, we do have placentals. Some of them were the kind of placental style of our tritiality. Some of them were precocial. We do seem to have that precociality established well before the KPG boundary. But it might actually have been ancestral now. Instead of being this derived placental exceptionalism thing, it might actually be something that they inherited quite early on in mammal evolution, if this is not an example of convergent evolution with these multituberculates. In contrast, the marsupials did indeed seem to have their marsupial style maternal investment strategy established before the boundary, and that may have played a role in their extinction dynamics at that boundary. Now, expanding on this is the topic of an NSF proposal that I've been banging my head away at for the last couple of weeks. And so we're hoping to get some more sampling within these other lineages and go even further back on the stems to really polarize these characters and see what the ancestral states are here in mammal evolution. And to me, the work that I'm currently doing is laying the groundwork for a long-term research program that talks about investigates the evolution of mammal life history and physiology. So one of the first of my next steps will be to continue building the empirical framework for this dental trace element mapping technique based on living species. So far, this dental trace element mapping technique, the heat maps, have only been applied to placental mammals, and even then only a couple different lineages, things like ungulates and primates. And so it's really important to sample broadly among living taxa with known nursing histories to see if we get the same sorts of signals and how they manifest in species with very different life histories. For example, we might expect, based on our knowledge of their development, that the life history of marsupials would leave a different trace element signature in their teeth. Their teeth only begin developing after birth, so they shouldn't have that zinc signal that we see. And they have a longer suckling period with more variation in the chemistry of the milk through time, and this might affect their barium uptake. So I've developed some models for how this might work, but of course we need to ground truth these in the modern to be sure that we can actually evaluate these signals back in our fossils. Now, fortunately, I've already taken the first steps towards this because Toronto is chock full of urban opossums who have one way or another made themselves available for research, shall we say. And so I've gathered a micro-CT data set of these teeth developing through ontogeny using Luke Muller's excellent CT scanner. And I've made some thin sections of those exact same teeth that allow us to correlate the growth with the daily laminations with these bigger patterns in tooth development across the lifetimes of these organisms. And these show really fantastic enamel laminations, so they're now ready for trace element analysis to kind of give a descriptive picture of what opossums are doing chemically early on in their lifetimes. Some of the other next steps are looking at other ways of collecting data entirely. So I've been working with a couple other early career scientists, and we've been using synchrotron micro-CT data. So synchrotron is a super high-powered particle accelerator that produces incredibly powerful X-rays. And as soon as we saw results like this, I kind of swore under my breath because it means I might not need to make another thin section in my life. This is the bone of a Cretaceous eutherian mammal from Uzbekistan. And to give you a sense of scale, these are grains of sugar in between. And each of these little black dots here is a bone cell, except for some of these that are blood vasculature cells. But we can see all sorts of different textures within this bone. And I wasn't able to attend because I had to come back for teaching here, but my colleagues had another very successful session at the Diamond Light Source in the UK where they were looking at the teeth specifically to see if we can get those enamel laminations in the teeth non-destructively. I've also been working a little bit more in the modern, trying to apply some macroevolutionary techniques to sort of understand what the pattern and pace of life history evolution in living mammals is, where we have a much better sense and a whole lot more data. What's really exciting about this work is that we're taking some of these trees of mammal evolution and we're finally able to root them with fossils for which we can have the same scale of data. So we can incorporate these fossils with a gestation period that matches the scale of our living animals, and that is really helping to polarize the ancestral state estimations. My colleague, Theo Danis, is working on some advanced techniques, which I don't necessarily fully understand all the math behind them, but to try and tease apart this complicated network of life history traits and how they interact and govern each other. And beyond mammals, I'm also interested in seeing whether dietary trace elements might be preserved in reptiles, like dinosaurs. One challenge, of course, is that reptiles constantly replace their teeth, and so we need to sample many different individuals of different sizes in order to get the same sort of longevity analysis. And as far as I know, reptiles also don't drink milk, although Sicilians, I guess, produce milk, but we don't know chemically how similar it is to mammals. But fortunately, there's a lot of promising evidence from other lines, other stable isotope lines like zinc, that might have an opportunity to reflect trophic level. And so if we can map zinc isotopes in the same way we're doing with trace elements, we might be able to see when organisms are changing their trophic levels throughout their lifetimes, which might give us some sense of parental care. And in the same vein, I'm interested in investigating other trace element or isotope systems for ecology to see if we can map these changes with these daily chronologies and understand how the biology and ecology of animals are changing through their lifetimes. And to me, these next steps all work towards a broader goal of understanding the evolution of life history and how it has shaped evolution itself. So I want to look further back across the tree of mammal evolution. I've started in the Paleocene with the data on Pantalambda and those other Paleocene animals to get a good sense of how they look at this early stage of their evolution. And I'm filling that picture out about the extinction by looking on the other side with these three groups to see how they survive through this boundary. And next, I would like to look further back into the Mesozoic. So I've had commitments from now 10 different institutions around the world to sample their material using these techniques. And this covers basically the entire Mesozoic. So we've got all sorts of different mammals and mammal ancestors in there. And I'm working to continue expanding this sample. And hopefully what we can do is quantify and categorize life history styles in these fossil mammals to give us a better understanding of when and where these different physiological and life history traits evolved in these organisms. And then use some evolutionary modeling techniques to see how did this actually affect things like diversification rates. Can we predict which lineages in the past might have radiated because they developed these new reproductive innovations and things like that. And especially working towards understanding how life history has affected extinction rates in the past. And that's, I think, maybe one of the things I'm most interested about is using this research to look at deeper links between life history and environmental change. There are certain model ecosystems like ancient Alberta that offer an opportunity to track and trace the community structure in response to these different environmental signals that we have preserved in these same rocks. So these late Cretaceous rocks not only preserve these amazing fossils, but they also preserve evidence of the environments through time via all sorts of different proxies. And that stratigraphy, in many cases, is resolved on sub-million year scales. So we're starting to approach some of the scales we would need for understanding conservation in the modern. And together, these kinds of systems let us see these signals of major changes in climate, sea level, precipitation, all sorts of other environmental things preserved right alongside the fossils. And if we can get daily information about life history from the fossils, we can tie it and start asking some big questions here. So for example, did the different groups of mammals in the Cretaceous invest like today's mothers do? Do we see any trends in life history across time, like building towards longer pregnancies? And finally, we can ask how mammals responded to the environmental changes that we actually see here in these rocks. And to me, this final question is most important, especially on a day like today, Earth Day, because it helps us understand what changes we might expect in mammal communities today as we face climate change and biodiversity change. Projects like this one help us to understand how rapidly mammals can respond to environmental change and disruption, and how they might respond to changes like the current climate crisis. The fossil record is really the only way to study animals and ecosystems over long time periods. And so paleontology, despite seeing it a little bit removed, is actually crucial for understanding the long-term changes that are going on around us right now. To me, the final exhibit at the Terrell Museum of Paleontology in Drumheller says it best. Understanding ancient life and the circumstances in which it flourished or died helps us recognize our place in nature and how our decisions affect all life on Earth. And with that, I have a whole bunch of people to thank. I've received a lot of support for my research from various institutions and funding sources, and a lot of intellectual contributions as well. This is certainly not something I have done on my own. And I really hope that I've interested you, and hopefully I've spurred some ideas in you, and I'd love to hear any of your questions about my work. Yeah.
I'm just blown away. That was so cool.
Oh, thank you.
Yeah. I don't even know what to ask because it was just all incredible. But I guess I'm wondering, like, I think I wasn't aware that you could just get this much information out of any fossil. And I guess in my experience, coming from more of like evolutionary genetic side of things, like, you know, when you're making a phylogeny, there's fossils that you use to sort of calibrate things, but you go look at the literature and it's kind of confusing where that actually fits in the tree.
Yeah.
Like, what is the sort of potential of these really fine scale methods of accessing, like, I don't know, like leaf stomata, like really fine scale traits that could be informative for placing fossils into existing trees across like many organisms, and how easy is that going to be for any biologists to like start doing that?
It's a big undertaking. So partly there's a couple different aspects to this. Firstly, it's not your fault that you didn't realize people can get this info from fossils, because nobody did really. We've known about these daily increments in teeth for more than a century, and only 2 or 3 papers have tried to apply them to fossils. So it, for whatever reason, is underappreciated in these animals. And the geochem stuff, we really didn't know that this was possible. And we don't even really know the boundaries of it yet, of how much data we can get. But I think this is part of this wave of paleontology that is inspired more by evolutionary biology. And I almost want to call it paleobiology instead of paleontology. That's a term that a lot of paleontologists have started identifying themselves as, because there's a shifting focus from documenting the fossil world to understanding how fossils contribute to our understanding of evolution and biology. And I think that's this wave of young scientists starting to do this kind of stuff and saying, all right, how far can we push the envelope? How much data can we get out of these? And I'm happy to say that as cool as this work is, I think there's far cooler work out there in terms of getting all sorts of other signals from fossils, even down to, kind of molecular remnants and how that affects physiology. So there are some people looking at metabolic signals in fossils that are 300 million years old. And to me, that's really pushing things in ways we didn't know they could go. So there's a lot of work to get these onto a level where they can be shared and implemented and used by evolutionary biologists as though they were living animals. And that's why I'm really excited about my collaboration with those scientists at Vanderbilt University, because they are trying to do that. They're looking at these, big picture macroevolutionary questions and starting to fit in these fossils. A lot of these mammals are represented only by their teeth. And so this technique is really nice for being able to get data out of those teeth that are quite abundant. But at the same time, that leaves a lot of confusion about their relationships. And that's especially true for the Paleocene. So Pantolambda is a eutherian, basically in Cetus. We don't know where it goes within the eutherian tree. And I'm working with some colleagues who are using molecular and morphological techniques to try to clarify those relationships early on. But when you have irradiation and organisms diversify morphologically after they genetically diversify, it can be really tough to use morphology alone to reconstruct what's going on in the tree. So those kinds of things, I mean, partly too, it's a lack of integration. It's me as a paleontologist not being trained in the methods you're using as a geneticist. And so I don't know what sort of information we can share. And that's why I think opportunities like this are great, because it gives us a chance to get face-to-face and see what each other are doing and how we can use it.
Yeah. So I have a question, and I'll preface it by saying, by the way, also, this was a very fun talk to hear, but I know nothing about mammals, or very little about mammals, and very little about fossils and macroevolution. So, but among people who study contemporary life history and demography, there is as much emphasis on survival and how long an organism can live as on reproduction and how, many offspring an organism has or how well they live. And you talk mostly about the speed and pace and independence of reproduction. Is that because we know that for mammals, it's all about reproduction? Well, actually we don't in contemporary demography. We see the same pattern as we do with everyone else. Or are there techniques to think about longevity and aging?
There absolutely are. Yes. So when you look at, if I go way, way back, beginning, these growth marks are annual. And so in an organism's skeleton, they do give you an approximation of how long that organism lived. There are similar structures that are also on the roots of the teeth called cementum annulations. And basically the cementum is part of the periodontal system that anchors the tooth to the jaw. And so it forms as part of that tooth socket, and it basically incrementally mineralizes the periodontal ligament through time. And so you get these bands that actually do reflect the longevity of the organism. So if I zoom forward a little bit, one of the aspects that we did analyze here is, we called it lifespan instead of longevity, because of course there are some slight differences there. But we were able to derive lifespan estimates for this animal. And what we found actually is that it's living on a very fast timescale. So the oldest individual that we found was 7 years old, which is less than half of what we would expect for an animal of that body size. But it comes with its own challenges, because very few organisms in a population reach the maximum lifespan, and even fewer of those will actually be fossilized. And so we're our sampling is not great enough to actually resolve what these lifespans are. But a colleague of mine is using, basically, he's ameliorating that issue by using synchrotrons. So you don't have to destructively sample the tooth. And he's sampling huge numbers of things. And what he's found is that you can get some of those lifespan trends in time still embedded in the teeth. So for example, early proto-mammals, things called morganeucodontids. They're these tiny little, like honestly that long, the whole body, like on the order of grams of mass. And they're living 20 years based on these teeth. And so those kinds of signals are substantially different than what we'd expect for an animal of that size today. So there are some questions to do with that. And this is a part of my work. It's something I didn't focus on today just because there's so much in here about reproduction. And to me, I think that's maybe the more innovative part of my work, is looking at reproduction, because that has been a big black hole in mammal evolution. As far as paleontologists go, we still don't know when mammals started to produce milk. We don't know when they stopped laying eggs in whatever lineages did. So, but yeah, lifespan and longevity are absolutely things that can be studied in these fossils, and maybe even a little bit easier than some of these reproductive aspects. Yeah.
Couple of questions. One is, so does this sort of thing also manifest in bone, or is that whatever? And if not, what is the feature of the tooth that captures and hangs on to the signal for 100 million years?
So it's likely that the signals do initially result, or they are also manifested in the bone early on in life. The difference is that bones remodel themselves through life. And so the earliest record of growth is actually erased in almost all organisms. Bone is this kind of dynamic tissue that repairs itself, and as it does, it eats away the earlier material and replaces it with new stuff. Yeah. And so these signals early on in life are usually destroyed by the time you get to adulthood.
I guess what I'm more just asking is whether these not necessarily the transitions and what you're being fed, but some kind of a signal that would be present in a fossil bone.
Yes, so the other difference is in the content of organics. So enamel has much more hydroxyapatite percentage. It's about 98, 97% hydroxyapatite. And that hydroxyapatite doesn't seem to alter during fossilization. Even the dentine of these teeth often does show replacement with diagenesis. And that's because it has a higher organic content that gets kind of leached out and then replaced. Bones also have the same sort of thing going on. Being very porous with all those vascular spaces, they absorb a lot more water, and their permineralization process incorporates a lot more new things than enamel does. The enamel basically is the same enamel as when this organism was living. If anything, there's been like 2 to 3% change, whereas in the bones, there's a lot more change that goes on. So partly it's the signal would have been there, but diagenesis, that process of fossilization, is taking those things out, or more likely it's overprinting those signals, making them a lot harder to detect. Yeah, exactly.
We have a couple questions online. Both thought it was an awesome talk. So there's both of them kind of regard think about other life history changes like ontogenetic diet shifts that could be picked up using this method and through other trace elements. So have you thought about doing that with diet shifts or periods of like hibernation or periods of resource scarcity that organisms experience?
Yeah, both very cool questions. I think there is an opportunity to do that, especially with some of those isotope systems like zinc. Zinc 66 to 64 as a ratio reflects trophic level, at least in marine systems. And the idea is that it probably also does in terrestrial systems, but because there are fewer levels, it's less expressed. There are some difficulties in getting those trace elements, which require a slightly different type of multi-collector ICP-MS. So some of the chemistry is slightly different. In order to get the precision for an isotope ratio, stable isotope, you need to use a different machine that doesn't allow you to control diagenesis the way we have. So it would take kind of an extra step in the approach to get there, but it is something that I would like to do eventually. In terms of those other ontogenetic changes happening later on in life, we're limited in some ways by the enamel, because the enamel only forms for an early window while the tooth is forming. Once the tooth is in place and it's started to erupt, the enamel doesn't form anymore, which is an advantage for maintaining the signals, but it's basically a limitation in terms of how long that window is. And as the tooth wears down, you start to lose that record through time as well. But cementum, which has those yearly bands, can actually preserve some of these signals as well. And there's good studies on modern organisms looking at changes in cementum chemistry through time that do seem to reflect changes in lifespan, not lifespan, in ontogenetic diet over the organism's lifespan. But there's a couple limitations. Cementum is more similar to bone in terms of its chemistry, and so you get some diagenetic overprinting. The resolution as well, the cementum is usually very, very thin compared to these other structures. And so the bands themselves contain less temporal information. You could look over a long time scale, but you're kind of limited either by looking at a longer window with less precision or a more precise but much shorter window. So I think those kinds of questions would actually be better addressed with reptiles, because reptiles constantly shed and replace their teeth throughout their lifetimes. And so if you can have good control for what age of a specimen the tooth came from, using, you know, maybe just using skeletons that actually give you a sense of size of the organism, then you could potentially look at these things a little bit more robustly than mammals. So trade-offs and advantages both ways.
Oh, Nick Peoples has one more question, and then we'll cut it off with that. Wants to know about polyphyodont species. I might be butchering the
No, you got it right.
pronunciation of that. Would the lifespan of the tooth be too short to pick up the major changes that you can see in mammals, or could the continuous replacement actually help in identifying changes later in life?
Yeah, great question.
I
and
guess Denny said maybe you answered first.
Yeah, so polyphyodonty is the process of replacing your teeth multiple times throughout your life, in comparison to diphyodonty, which is what we have, just 2 generations. Unless you're a hockey player, and then you get dentures after, so you get the thirds. But polyphyodont animals, yes. As far as we know, the window of time that it takes a polyphyodont animal to produce its teeth is subannual, so it's quite short. In things like crocodiles, it can be like 30 to 60 days. But in some of the dinosaurs, it can actually be substantially longer because they have much larger teeth. And there have been good studies about replacement rates of these teeth through time. And I don't know the figures off the top of my head, but I would imagine there are probably some species for which you could get lifetime changes from a single tooth. But more likely what you would have to do is sequentially sample a population of different aged individuals to try to get kind of a generalized signal out of all of them. So one of the projects that I'd like to do, I've done some work on baby tyrannosaurs, where we've got these like embryonic tyrannosaur jaws, and we've got some teeth that are just a little bit bigger and some teeth that are just a little bit bigger than that. And looking at those to see within a single kind of fossil formation, are there differences in the trophic levels between all of those? And if there aren't, that might be an indication that there is some evidence of parental care in some of these animals, because if a baby tyrannosaur is eating the same thing as an adult tyrannosaur, it's certainly not hunting triceratops. And so that might be evidence that it's getting fed in that sort of way, because I would imagine that a baby would actually have a much higher trophic level because it's eating things like lizards and insects and stuff that are kind of a little bit higher than the primary consumers. So that might be one way that that reptiles could help. It's just, it needs framing the question a little bit different way because of how polyphyodonty works, but advantages and disadvantages to each.
Great.
Awesome.
All right.